here together with us. Can you hear us? Can you drop us a message if you are?
Hello, everybody.
Let's see. Okay, perfect. Yes. Okay, so hello everyone, and welcome to this today's session of the Brain-Body Interactions virtual seminar series. We may wait a little bit more to see if more people join in. Just a couple of minutes, and then we'll start. And I'll take these minutes to say something that we've been thinking. So on the behalf of the committee behind this series, we are very, very happy about the community of people that we've been bringing together, and that is growing, and that it keeps growing. This series started as with the goal of bringing together scientists interested in bridging the gap between neuroscience and other biological fields. And if you have been with us for the past weeks, you know that we have been having some super interesting talks covering many scientific fields from brain to body. So some of you already voted, and some of you already saw it. And today we have this poll. And so we want to know more about you. We want to know more about the people from this amazing community, and actually, where do you think you would position yourself in this brain-body spectrum? And so we encourage you to participate during the talk on the poll that is available if you click on the polls button down, and vote for the field that you would position yourself, and we'll get back to this in the end of the talk, and we can have a little chat also with Isaac to see his opinion about it. Okay, so let's wait a bit more. For those of you that are new here on Crowdcasts, if you want to ask a question during the talk, you can press the Ask a Question button down, and either ask or vote for an already asked question. And I think, okay, so I'll also leave here on the chat the link for you to subscribe to our mailing list if you don't want to miss out anything from this series. It's a super quick form. And let's see.
We are all interdisciplinary. That's right. The body and organism, right? No, I think it's great that there are people coming from different perspectives.
Yes. Yeah, I think it's very. I think this poll will give us a little bit of notion on where everybody's standing. And okay, so I think maybe I'll start.
Sounds good.
So welcome everyone today again, and welcome Dr. Isaac for here on this seminar. It's my pleasure to have you here today and to host you on this virtual seminar series. And so I will start by present you. So Dr. Isaac obtained his PhD in immunology at Harvard Medical School, working in Dr. Mike Carroll's lab on the role of the microglia and T cells in AL. He did his postdoc in Dr. Tom Maniati's lab in the same research area. He moved to study the field of neurobiology of pain, and for this he worked in Dr. Clifford's Wolf lab at Boston Children's Hospital, where he studied the role of nociceptor neurons and pain in bacterial host defense. And currently, Dr. Isaac Chiu has his own research group at Harvard Medical School in the Department of Immunology. and his lab is focused on the neuroimmune interactions in pain, itch, and infection, and how the understanding of these interactions could potentially lead to novel approaches to treat pain and inflammation. Aside from this, Dr. Chiu has received several recognitions, including the NIH Director's New Innovator Award and the BWF Investigators in the Pathogenesis of Infectious Disease Award. So with this, I would like to thank you again for accepting the invitation of being part of this series, and the stage is yours.
Thank you so much, Raquel. That was a really nice introduction, and it's my honor to be here. I really love this seminar series, this topic of brain-body interactions. I think it's wonderful that we have neuroscience, immunologists, and people from different perspectives. But as one of the comments said, there's should not be boundaries, because I think biology is interdisciplinary. And I think the topic that I'm going to tell you today is really combining perspectives from immunology, neuroscience, and microbiology. So I'm going to start to share my screen now, and I hope that you can all see it in a second.
Okay.
Okay, can you see it?
Let me just change it. Okay, yeah, it's perfect.
Okay, great.
I think it's, yeah.
So today I'm going to tell you about neural immune interactions in pain and host defense. So first, just some disclosures. Okay, so for my talk today, I'm going to give you an introduction on pain and immunity, and then talk to you about two different places where pain and the immune system really intersect in the context of actually infection. One is in the skin, and I'll tell you about how nerves signal to immune cells in the skin during a really invasive form of bacterial infection called necrotizing fasciitis. And then the second one is in the gut, how pain fibers in the gut talk to the microbiome and also the epithelial cells to protect against salmonella invasion. So all of us have experienced pain, and what has it always been linked to? Actually, even from 2,000 years ago, Celsus, the Roman physician, linked pain to inflammation. So it's one of the four cardinal signs of inflammation. So redness, rubor, heat, calor, tumor, swelling, and dolor is pain. And it's increasingly clear that pain is not just a symptom of inflammation, but actually the neurons that mediate pain actively participate in the inflammatory process, in mediating and modulating or driving it. So I just want to point this out, that pain and the immune system, which we think linked to inflammation, has, in a sense, always been linked in a way. So now what about pain? We know that pain fibers, or the nerves that mediate pain, are really densely packed, particularly in border tissues that interface with the environment. For example, the skin and the gut, these are actually some stains of pain fibers that some students in my lab took. And you can see the really amazing density of these nerves in the glabrous skin of the mouse, as well as in the GI tract. And you can imagine why they need to be so dense in those areas, because that's where the tissues are most exposed to the environment and to potential dangers. And so pain is actually transduced by specific types of sensory neurons called nociceptors. So nociceptors, it's a term that came from Charles Sherrington when he coined the term from this, thinking about neural reflexes. Pain is a classic neural reflex driven by neurons that sense noxious stimuli. So nociceptors sensing noxious stimuli. And they transduce these signals from, as I mentioned, the skin and other barriers, the joint, the genitourinary tract, GI tract. They take these signals from these peripheral sites via their cell bodies, which reside in peripheral sensory ganglia, such as the dorsal root ganglia, to the spinal cord, where then these signals are transduced via interneurons to the brain, and then it's processed as this unpleasant sensation of pain. And the fundamental role of pain is to protect us from danger. So we know that these nociceptors have specific molecular sensors at their nerve terminals that respond to increased temperature. So like trip channels respond to heat or cold, and also to reactive chemicals. So there's specific ion channels that can actually detect reactive chemicals, and also to tissue injury. So they can respond to mechanical force, as well as factors released, like damage-associated factors like ATP. And finally, our lab is finding that these neurons also can respond directly to microbes, which is another source of danger, pathogens that invade our tissues. And the response is that the nociceptor will then lead to classic avoidance behaviors from these dangers, so that we can avoid next exposures. But also at the cellular level, these neurons talk to the immune system and coordinate a response against danger. What about microbes and pain? So the theme of my lab is really to understand how pain and host defense interact. And so we know that during infection, there is pain is a characteristic of many types of infections. So just as some examples, when you have a Staph aureus skin infection, when you have an abscess, or strep throat, which is caused by strep pyogenes, or a gut infection caused by salmonella, these are all characterized by pain. And for a long time, it was kind of assumed that the reason you have pain is because it's downstream of immune factors or inflammatory, the swelling of the tissue. But it turns out, as I mentioned earlier, these neurons can also directly sense pathogens. And so this was work that I did as a postdoc in Clifford Wolf's lab, where we identified that Staph aureus bacteria actually can be directly sensed by these nociceptors via at least two different mechanisms. So formulated peptides, which are released by Staph aureus, bind to FPR1 on mechanical nociceptors, so nociceptors that mediate mechanical pain, as well as a pore-forming molecule, alpha-hemolysin, which can directly form basically large pores in the membranes of cells, including neurons. And this leads to cations coming into the nerve and then the firing of the pain fiber. So these are just revealing some of the mechanisms that neurons can sense pathogens. There's a growing area of research here that, like immune cells, these neurons can express toll-like receptors and other pathogen recognition receptors to produce pain during infection. In response, once they sense these noxious stimuli as well as pathogens, these nerves then can regulate the immune system. So this is actually what's called antrodomic signaling, where there is a nerve impulse that goes up towards the brain to mediate pain, but actually back propagates along neighboring axons of the same neuron, leading to the release of neurotransmitters and neuropeptides from the nerve terminals of these neurons. And these ones then signal to the immune system, whether it's to mediate or modulate chemotaxis, dendritic cells or T cells, or the vasculature to mediate actually what's called neurogenic inflammation. So the nerves themselves will induce swelling of the tissue. And this kind of neuroimmune signaling is just a really fascinating new area. And I think there's a lot of questions of how the nerves talk to the immune cells. So I just wanted to highlight that there's actually quite a lot of work, recent work that's really exciting on how nerves, peripheral nerves, talk to immune cells, including elegant work by Enrique Vega Fernandez's lab, as well as Vijay Kutru and David Artis's lab, recently showing that cholinergic neurons in the gut and the lungs release a neuropeptide called NMU, which regulates innate lymphoid cells in the context of worm infection and allergic airway inflammation. So this is another substance, not pain fibers, but these are cholinergic neurons that signal via a neuropeptide to immune cells. Also, enteric neurons can actually produce IL-18. This is work by Richard Flavell that can actually directly talk to goblet cells in the gut to mediate barrier function. And then Dan Musita's lab had very nice studies showing that sympathetic neurons, which are another branch of the nervous system, not pain fibers, but stress-related, that signal via norepinephrine to macrophages and ILCs to modulate their responses to infection. So these are all other types of neurons in the peripheral nervous system, and this is a really exciting area. Sensory neurons and nociceptors also release neuropeptides and talk to immune cells, and that's going to be the topic of my talk today. So what I've shown you so far is that neurons that mediate pain, nociceptors, can directly sense microbes, but there's big questions on how do they do that, so the molecular mechanisms. Secondly, these neurons can release mediators that talk to the immune system. So this is a really interesting area. And also what I won't talk about today is the immune cells can talk to these neurons via cytokines and inflammatory mediators. But how does all of this integrated into host defense? So I'll tell you again 2 areas where we think this is highlighting this sort of tri-interactions going on at the barrier. So the first one is work that was done by Felipe Ribeiro, a very talented postdoc in my lab, in collaboration with Mike Vessels at Boston Children's Hospital. And this is on the role of pain and neuroimmune signaling in the skin in host defense. So in this context, we were studying strep pyogenes infections, group A strep. So this is a bacteria that's commonly colonizes the throat and causes up to 600 million cases a year of strep throat. So that's a painful sort of inflammatory swelling of the throat. But it also causes a whole panoply of other types of infections, including very invasive infections of the skin and soft tissue like cellulitis and necrotizing fasciitis. And we were very interested in this because pain is such a big part of particularly invasive strep infections like necrotizing fasciitis. So flesh-eating disease is the common name for this really terrible form of infection. And it's highly invasive and often very difficult to treat. The strep pyogenes is a leading cause. So Mike, who is our collaborator, is a clinician, and he noticed to us that said that, you know, diagnostically, one of the early symptoms of flesh-eating disease is pain out of proportion, so really intense pain without much other signs. So the question started to ask, you know, why is this sort of infection so painful? And then, do these nerves regulate infection? So how do we study pain? So this is for those of you who are interested in the kind of nervous system part of things. Well, there's some very acute assays where we can measure nocifensive pain. So these are kind of behaviors that protect the mouse, you know, showing kind of acute pain responses. And here what we've done is we've infected the mouse in the foot pad and looked at these responses. And so you can see with the group A strep, you get this sort of lifting, licking response of the foot pad, as well as immediate flinching behaviors. So, you know, these manifest are pretty intense pain. So this bacteria in mice can cause significant pain, and we can quantify that by measuring these behaviors in the course of an hour. And it's in a dose-dependent manner. So the more colony-forming units we infect the mice with, the more of these acute pain behaviors we observed. And secondly, we also looked over the course of time of infection. So we used 2 different strep pyogenes strains, the M1 and M3, both of which are human strains that cause necrotizing lesions. And in mice, this leads to a significant swelling and damage to the tissues that is very invasive. And what we can also measure during the course of infection is what's called hyperalgesia or hypersensitivity, by looking at their response to mechanical stimuli. So this is called a von Frey assay, where we're looking at using filaments of different pressures to look at their response. So this mouse, for example, is responding, you know, we're measuring the number of responses they have to this filament. And that response goes up when there's, you know, hypersensitivity or inflammation. Similarly, we can look at heat responsive. So this is using a radiant heat source in this Hargreaves assay, and we measure the latency to their flinching behavior. So that latency drops when there's also inflammation. And so what we noticed was that with the infection with the group A strep or the strep pyogenes, increasing doses of infection also cause a very quick increase in this mechanical hypersensitivity. So you need a much lower threshold filament, 0.008 grams, to induce a response in mice post-infection at these early time points. So we've kind of modeled strep pyogenes infection in mice and can study their pain. And we were asking, why was it so painful? So it turns out the mechanism is via a pore-forming toxin the bacteria produces called streptolysin S. So this is a very important toxin produced by the bacteria that actually causes the red blood cell lysis if you put them on blood agar plates. And it's a polypeptide produced by the bacteria encoded by this SAG-A gene. And all of these other genes in this operon are involved in this post-translational modification secretion of this peptide. So we wanted to ask, because we had previously found that another pore-forming toxin from Staph aureus, alpha toxin, can directly activate pain fibers. So we wanted to ask, could it also be the case for this strep toxin? And indeed, it's the case. And I won't show you data in vitro where we saw neurons responding directly to the toxin. But in vivo, we showed this was necessary for pain, because if we used bacterial mutant strains that lack the SAG-A gene, which encodes the toxin, but not this other toxin, SLO, which is a different pore-forming toxin, this eliminates the pain during infection caused by both the M1 and M3 strain backgrounds. We also showed that this mechanical hypersensitivity that I mentioned is dependent on this streptolysin S or SAG-A. As you can see, actually the legends here are not clear, but basically the yellow line here and the black line are the ones that lack the SAG-A gene, but not the SLO alone. So this toxin from the bacteria is inducing pain during infection. So we wanted to ask, what is the consequence if we remove the pain-sensing neurons on the actual infection itself and immunity? So the way that we targeted neurons is to use the ion channel TRPV1, which is a well-known nociceptive ion channels expressed by a large proportion of nociceptors, and it binds to capsaicin, which is the burning ingredient, pungent ingredient, chili peppers, that causes that painful response. But endogenously, it also responds to protons and to noxious heat. So we used it as a marker for pain fibers. So we have TRPV1-driven CRE, where we've crossed that with the diphtheria toxin reporter mouse. So this will, during development, ablate or kill off all the TRPV1 positive nociceptors. In the control litter mates, which lack the CRE, the nociceptors are intact. So what we found is if we infected mice with strep pyogenes, that we actually saw the nociceptor deficient mice having a stronger immune response. So this is by flow cytometry, where we found that they actually had more neutrophils in particular coming into the skin in the TRPV1 neuron ablated mice. As a second approach, we also killed off these neurons using a chemical called resnifratoxin, which binds really highly to TRPV1 and causes the calcium overload in these neurons that leads to their loss. In both TRPV1 CRE DTA and RTX mice, we saw increased neutrophil numbers coming into the skin. And this actually helped clear the bacteria. So this was very interesting. So we were asking, how could the neurons regulate neutrophils in the skin? So I had mentioned that one of the ways these neurons communicate with immune cells is via neuropeptides stored in their nerve terminals. And one of the key ones that nociceptors express is called CGRP. And CGRP binds to its receptor called RAMP1, which is a G protein coupled receptor expressed by actually many immune cells, including neutrophils, macrophages, and dendritic cells. And however, the role of CGRP in neutrophil function had not been previously looked at. So we wanted to see, does CGRP regulate neutrophils? And indeed it does. So what we found is that if we took either mouse bone marrow neutrophils or human blood, which contains neutrophils, and we mix them with strep pyogenes, we can do what's called a Lancefield assay, which is incubate them and look at the multiplication of the bacteria in the presence of neutrophils. And normally the bacteria gets either stopped from growing or killed by the neutrophils. But if we add CGRP to the mix, it actually inhibited the neutrophil from killing. And this was true as well in the human whole blood killing assay. So we thought CGRP could be blocking the immune response. So can we find a way to block this neuroimmune crosstalk? So it turns out that there is a commonly used way, at least for skin, to inhibit neurotransmission, which is Botox or botulinum neurotoxin. So the way botulinum neurotoxin works is that it cleaves SNAP25. So there are actually 7 different serotypes of botulinum neurotoxin. A serotype is the one that's used clinically, and SNAP25 is a very specific snare protein in neurons. So if you cleave that, it will block vesicle release from both nociceptors as well as other neurons in the skin. And the second thing that we thought about was using an antagonist against this particular neuropeptide called CGRP. And the CGRP antagonists have gained a lot of attention recently because it's actually a critical mediator of headache and pain and migraine. So could we repurpose one of these CGRP antagonists that's being used for migraine to enhance the immune response during infection? So this is what we did. We treated mice with the Botox or with the CGRP antagonist and then infected them with the strep pyogenes. And we found that in both cases, this increased neutrophil recruitment into the lesion site, as well as resolved infection. So here what we're doing is we switched from the foot pad to the flank to measure the size of the lesions over time. And you can see the bacterial vehicle-treated mice have these really large dermatocrotic lesions that occur with the strep pyogenes infection. But the mice treated with either BONT-A, the Botox, or the CGRP antagonist had much smaller lesions that resolved much quicker. So what I've shown you here in this very first story is that there's a bacteria that produces a toxin, streptolysin S, which causes a lot of pain during infection. And this actually helps the bacteria because at the same time, the neuron releases a molecule called CGRP, which inhibits neutrophil recruitment and neutrophil killing of the bacteria. So it's exploiting a pain neuroimmune circuit locally that can help the bacteria survive. But we can kind of short circuit this neuroimmune suppression by using either botulinum neurotoxin or a CGRP antagonist, and this could enhance the immune response locally. So we're very excited about this because it could be potentially used to treat either necrotizing fasciitis or other types of skin infections that involve neutrophils and pain. So that's something we're pursuing. Okay, so then I'm going to tell you now, switch gears to a different tissue. So the pain fibers I mentioned in the skin are involved in immunity there. What about the gut? So we think a lot about the GI tract and the gut-brain axis, how bacteria in the gut can regulate the nervous system. What about the opposite direction? Do gut innervating neurons regulate the microbiome in the gut and also host defense against invading pathogens? So this was work done by Nikki Lai, who was a graduate student in the lab, and she defended her thesis actually last year. So she's now actually a scientist at Calliope Pharmaceuticals. So this was published recently. And what we wanted to ask was, what are the roles of nociceptive neurons? The same neurons I mentioned in the skin that mediate pain in the gut mediate also pain, but visceral pain. And how do we target these neurons? So we use 2 different approaches. One is the same ion channel I mentioned earlier, TRPV1. And a second one is an ion channel called NAV1.8, which is a voltage-gated sodium channel involved in generating action potentials in the beginning of a pain response. So nociceptors require these sodium channels to fire action potentials. And actually, there are mutations in this ion channel that leads to congenital deficiencies in pain as well as increases in pain. So why did we use 2 different markers? Well, these largely overlap in gut innervating neurons, and also they're complementary approaches to see whether nociceptors are involved in host defense. But before I get into these specific techniques, I want to give you a brief overview of the neuroanatomy of the gut in terms of sensory neurons. So the gut is one of the most complicated organ systems in terms of innervation. So there are sensory and motor arms. But even within the sensory arm, there are actually 3 different types of sensory neurons. So there are the sensory neurons that actually live within the gut itself, so the enteric nervous system. And these sensory neurons are these blue guys that basically reside in submucosal or myenteric plexus, and they mediate peristalsis, so they signal to motor neurons, and they also detect luminal signals. So they are self-contained within the gut, the intrinsic enteric neurons. Then there are 2 types of extrinsic sensory neurons. So one that live within the nodose jugular ganglia, and they project from more of the upper GI tract to the brainstem via the vagus nerve. So this is a real gut to brain signal. And they transduce signals related to both noxious stimuli, so related to, like I mentioned, nociception, as well as nutrient status of the gut, as well as stretch. And they can mediate reflexes such as nausea, vomiting, but also appetite and satiety. Then there are the dorsal ganglia neurons, which are more innervating the lower GI tract, and they take signals from the colon and the ileum and go up to the spinal cord, and they mediate pain. So I told you earlier about these pain-mediating nociceptors. So the 2 strategies we use to target extrinsic, they're very specific to extrinsic neurons. So they don't target at all the enteric neurons because these ion channels are absent in enteric neurons. Okay, so then the question we wanted to ask is, again, focused on a particular pathogen. We were interested in how nociceptive neurons regulated salmonella infections. So salmonella is a major gram-negative pathogen, causes up to 1/4 of diarrheal diseases. And it's, characteristic of invading the GI tract is very, very well characterized. So Salmonella typhimurium is the strain that we used, and it causes typhoid-like disease in mice. So in humans, certain Salmonella strains cause gastroenteritis, and other causes more of a typhoid-like disease. And typhoid-like disease is in this kind of step where you get a colonization transiently of the small intestine, ileum, and then these bacteria invade the Peyer's patch. So these are specialized lymphoid organs in the gut, specifically in the small intestine, and they invade via these specialized epithelial cells called microfold cells. And then they disseminate via myeloid cells, which take them out of the gut into the systemic circulation, and then you get typhoid fever and things like that in humans. So in mice, this causes these 3 steps. So we wanted to ask, are nociceptors involved in host defense at any of these 3 steps against the bacteria? So how do we approach that? Well, one of the strategies is using the NaV1.8 as a marker. So NaV1.8 Cre, just like with the Trivi1 Cre earlier, we've now used that mice to ablate NaV1.8 lineage neurons. So we've crossed this NaV1.8 Cre with the Rosa 26 diphtheria toxin reporter. And so then the Cre positive mice lack the neurons, whereas Cre negative mice are controls. So just to show you the ion channel itself, the gene expression is completely ablated in these mice in both the dorsal ganglia and the nodose ganglia. It's not present, as I mentioned, in the gut myenteric plexus. And this is confirmation of that. So marking DRG neurons with the CGRP, a large proportion of these neurons are ablated, but there's no effect on sensory neurons in the myenteric plexus labeled by calretin. So what happens if we infect these NaV1.8 Cre DTA mice with salmonella? So the protocol we use is orally gavage salmonella into the mice, and then measure the invasion at early time points in the ileum and the Peyer's patch at day 1, and then spread to the liver and spleen and other organs and weight loss at day 5. So what you see here is that the mice lacking the nociceptor neurons did poorly compared to the control litter mates. So there's log-fold more bacteria at day 1 in the ileum and the Peyer's patch in nociceptor ablated mice, as well as more bacteria in the liver at day 5, and also significantly worse weight loss. So as a complementary approach, we use the TRPV1 based approach. Here we have a TRPV1 diphtheria toxin receptor expressing mouse. So here we can, again, ablate the TRPV1 lineage neurons by injecting mice with diphtheria toxin or control with vehicle. And just like with the NaV1.8 approach, the mice lacking TRPV1 neurons have significantly worse off infection. So more bacteria in the ileum and the pyrus patch at the early day 1 time point, as well as more in the liver at day 5 and worse weight loss. So this is still not specific enough because both of these approaches will kill off both vagal and DRG neurons. So we thought of a different approach to just target vagal or DRG neurons. One of them, which is to target DRG neurons by intrathecal injection of RTX, which is this high potency TRPV1 agonist. And this actually leads to the loss, chemical loss, of the DRG neurons expressing TRPV1 only in the DRG, because that's where the DRG comes from, the spinal cord, but does not affect the vagal TRPV1 neurons. So he said, do these mice also have difficulties combating infection? And indeed they do. So just like with the total TRPV1 knockout neurons, intrathecal RTX, also these mice have difficulty clearing the bacteria. So more bacteria in the ileum and pyrus patch at day 1, and more in the spleen and liver at day 5, and worse weight loss. So we think it's a DRG neurons that are critical, because I won't show you the data, we also eliminated vagal TRPV1 neurons, and they did not play a role in salmonella infection. So how do these neurons play a role? Well, one thing we found was actually the neurons can directly sense bacteria. So the way we test this is we perform calcium imaging. So here we're growing these dorsal ganglia neurons, which include the nociceptors, and then we added Salmonella typhimurium to these neurons and did calcium imaging. And we could see there are subsets of neurons that responded directly to the bacteria and its contents. And then we added capsaicin, which activates TRPV1. So this labels all the TRPV1 expressing neurons. And then KCL, which will activate all neurons in the field. And what we noticed was that a large proportion of both total neurons as well as TRPV1 responsive neurons can directly respond to salmonella. So these neurons can detect something within the bacteria and then regulate something that mediates host defense. So what could that be? So Nikki looked at quite a few different potential mechanisms, including dysregulation of immune cells in the gut. And it turns out one of the key mechanisms is actually related to the microbiome. So as I mentioned earlier, the gut-brain axis has mostly been studied in the context of how the microbiome affects the nervous system, but the other direction, less is known. So we wanted to ask, could the nociceptors regulate the microbiome composition? And this could be important for host defense because the commensal microbes are critical for defense against pathogens, including salmonella. In particular, the commensal microbes compete for the niche, but also they can compete for nutrients for the pathogen and also stimulate protective host responses. So we looked within our nociceptor deficient animals versus control litter mates for microbiome related changes. And so Nikki performed 16S sequencing, and she looked across different parts of the gut, duodenum, ileum, colon, and also the feces. And within each one, she looked at the luminal contents versus the mucosal scrapings. And the striking change that we saw between nociceptor deficient and control mice was in the ileum, and particularly this red Clostridiaceae you can see here, where in the control litter mates, there's a lot of them, and they're specifically deficient in the nociceptor ablated mice. So the ileum also is where the salmonella invades the host. So this could be important. So we zoomed in to see what the differences are within this group. And it turned out the major difference was in a resident bacteria called segmented filamentous bacteria, which resides within the ileum and actually attaches to the epithelial wall And they form these, you can see these really cool, long filamentous forms. And SFB are very important because they are stimulants of host immunity. They stimulate T cell responses, in particular Th17 cells. And this is elegant work by Dan Littman Labs and others. And this is just, Nikki then looked through several litters of animals, Cre positive and negative, and you can see by qPCR, SFB really is significantly deficient in nociceptor ablated mice. And we also looked by electron microscopy because, as I just shown you, there's these really very clear forms of these bacteria. And what we found was that both in the villi as well as in the Peyer's patch, which is the site of Salmonella invasion, the dense carpet of SFB that you see was largely lost in the nociceptor ablated mice. So nociceptors are in some way there to maintain this microbe and its presence. So we wanted to ask, could they be involved in our mechanism? So what Nikki did was, in collaboration with Jun Hu's lab, who studies SFB, did fecal transplants of SFB feces or control self feces from the Cre positive or Cre negative mice into Cre negative and Cre positive mice. And what you can see here is the Salmonella invasion phenotype, where we see increased Salmonella in the nociceptor ablated mice, is somewhat restored when we defect in defense is somewhat restored if we put in the SFB feces. So this could be a protective mechanism. Secondly, we saw a very interesting crosstalk between the nerves and the M cells, which are these specialized epithelial cells on top of the Peyer's patch dome. So M cells, or microfold cells, were named that way because they form these indents on their surface, and they sit on top of these specialized lymphoid organs, and their role is to sample antigens from the gut to induce T cell and B cell responses. So that's their normal function. But they're also exploited as entry points for pathogens. So Salmonella, as well as other bacterial, as well as viral and even prion pathogens, utilize M cells as a vulnerable site of entry. So Salmonella actually uses its fimbriae to specifically bind to M cells via glycoprotein 2 on its surface. And we were wondering, could neurons be involved in talking to these M cells? Because we saw that there could be differences in the Peyer's patch site dome specifically. And so there was actually old papers, and this one was in a pig Peyer's patch by Volkanova et al., that found this really nice, beautiful network of nerves right underneath the Peyer's patch dome where M cells reside, including ones that express nociceptor markers like CGRP. So we asked, could they be interacting with M cells? So here, what we did is we stained for M cells using this glycoprotein 2, which is this surface protein of M cells. And we also stained for neurons with 2-1. And you could see within that dome region of Peyer's patch, this network of nerves, as well as M cells that you can see actually often juxtaposed to the nerves. So then we want to ask, could these nerves be regulating the M cells? So what we did is we looked in our nociceptor ablated mice that I showed you earlier, the NAV1.8 Cre DTA or the TRPV1 DTR mouse, and we quantified M cell density on the dome using GP2 staining. And you can see that the numbers of M cells actually go up. This is without infection at homeostasis in the nociceptor ablated mice, both per follicle of the Peyer's patch as well as averaged per mouse. So there are more M cells. The nociceptors are suppressing the density of these M cells at homeostasis. And I mentioned these are the gateways for the pathogens to come in. So this could be a key part of the mechanism, that nociceptors don't maintain these M cell numbers at lower levels, and therefore you get more invasion. And that is indeed what we found. So this is a complicated figure, but what we did is we killed off M cells in our mice that lack nociceptors, here ablated with RTX, with an anti-rank ligand. So rank ligand is a critical factor for M cells to differentiate. And what we found is that in mice that lack M cells, that also lack nociceptors, the problem with the host defense in terms of especially the Peyer's patch and the spleen and liver invasion is completely ablated. So M cells are downstream of the neurons. And so how does this work? Again, I want to come back to mechanisms. And so CGRP, again, is a critical neuropeptide produced by these neurons. And could that be playing a role in this M cell homeostasis? So what we did is we looked at CGRP expressing nerves. So CGRP here, there's two isoforms, calc A and calc B. CGRP alpha is the main one expressed by nociceptors. And so the GFP CGRP nerves were also closely juxtaposed to these M cells in the Peyer's patch dome. And what we found was that if we then took mice lacking calc A or CGRP alpha, they had density differences of M cells in the Peyer's patch dome. So at homeostasis, the M cell numbers are higher in the CGRP deficient mice, and also post-infection as well. So this is 5 days post-infection. Actually, Salmonella increases M cell numbers for its own sake. So it has a way of making normal epithelial cells into more M cells. But in both cases, the calc A knockout mice had more M cells. So what about the consequences on infection? Well, it turns out that these mice lacking this specific neuropeptide that's expressed by these neurons also have defective host defense. So you have increased Salmonella invading the ileum and Peyer's patch at day 1, and the spleen and the liver and at day 5. So I just wanted to summarize the second part. What I've shown you is that the Salmonella pathogen is one of the invasive pathogens of the gut, and the nervous system, in this case pain fibers in the gut, are critical to mediate the homeostasis of the gut. So that includes two things. One is regulates the densities of these epithelial cells that are entry points for the pathogens, so they shut down the numbers. And they also maintain the levels of a protective bacteria called SFB that reside on top of the gut. So if you don't have these neurons, the SFB numbers go down and the M cell numbers go up, and this allows in harmful pathogens. And you can imagine that, I mentioned this is happening at homeostasis, but let's say you have a painful stimuli, and these neurons could increase this process of shutting off the gates and helping, protect the gut from an invasion. So this is just in early days. We still have a lot of questions on how these neurons talk to epithelial cells in the gut. So summary of my talk. So the neurons I've shown you can directly sense pathogens to produce pain during infection. In the skin, I've shown you a story where the pathogen exploits this by inducing a neuroimmune circuit, which actually suppresses neutrophils. And blocking that using botulinum neurotoxin or CGRP antagonist can enhance the immune response. In the gut, pain fibers or nociceptors actually regulate homeostasis of the gut by regulating the composition of the microbiome and also the Peyer's patch M cells to protect against invasion. So there's a lot of really interesting questions of how neurons interact with immune cells and epithelial cells. And I think elucidating this crosstalk could lead to new ways of approaching inflammation and also pain. So, I do want to briefly mention some really exciting work that we've been doing also together with Yachi Chiu's lab who's a stem cell biologist. And we're really interested because we found that if we induce a lot of pain, actually with this RTX treatment in mice, the mice would consistently get gray hairs. So Yachi found that the way that this happens is that nerves in the skin talk to melanocyte stem cells in the skin and actually cause them to go into cycle. So they get lost and then you get gray hairs. So we're really interested in extending this I think rather novel question. How do neurons talk to stem cells? Both stem cells in the skin and stem cells in the gut. And could that also be involved in kind of pain and stress signaling in the context of host defense? So overall, what I've shown you here is that the nervous system is an active participant in host defense. So we have mechanisms that neurons can directly detect microbes, and that's an interesting area. If we can define those mechanisms, we could potentially block pain and other sensory signals. I showed you that neurons and immune cells communicate, and this could have a major impact on the immune response in these tissues against microbes. So there's really a lot of molecular questions at each of these interfaces that are very fascinating. And with that, I will close by acknowledging this was all really driven by two really talented people in my lab, Felipe Pino Ribeiro, who did the work on the skin and group A strep and pain, and Nikki Lai, who did the work on the gut. And it was together with wonderful collaborators, including Jun Hu's lab, Yachi Shu's lab. We have an interesting collaboration we're starting with Enrique and Steve Liberlees. And also we got help from experts like Vijay Kutru and others who are experts in immunology. So these are my funding sources, and I'm happy to take questions. Should I stop?
Thank you very much, Dr. Isaac. It was an amazing talk. We do have some questions here. So Abdul is asking, do we need to rule out that taking out of those neurons changes normal physiology of gut-like secretion?
That's a very good question. I agree that that is actually, we did in fact see some differences in gut motility. So that is a caveat of ablation approaches. So I think right now what we're trying to do is use chemogenetics to do more kind of activation or silencing. So it's not like eliminating the entire system. So yeah, I think when you eliminate any cell type in the body, there could be consequences on other physiological functions beyond just the immune system. So it's a definitely well taken point and good question.
Then Asia is asking, what may be the evolutionary rationale for the inhibitory response by the local neurons in the skin?
Yeah, so that's a very good question. So as Asia pointed out, we actually found that the neurons suppress neutrophils. So in the context of this type of infection that we saw with the invasive bacteria, that's very harmful because neutrophils are the army that clears the bacteria. But what we think is that in the context of other types of infections, like actually we're looking right now in the lungs at influenza virus and things like that, where neutrophils actually cause the damage. And other contexts like wound healing, where neutrophils can be very destructive, that's where the pain signal actually kicks in, where you have to have this kind of counterbalance against the immune response. So that's my kind of hand wavy explanation of why we saw that. But I think it's going to be very context dependent. So I think a lot of questions of how neurons, whether they drive or suppress immunity, why that's the case, in each situation.
Yep. So another question from Abdul is asking if those mutants also had reduced exudates. And he says mutants for toxins. I believe he's talking about the streptolizing.
Yeah, so we did see that they had less MPO, so myeloperoxidase production in the supernatants. So we didn't look at things like net formation, which could be very interesting. I think that, so in our study also with collaborator Brian Yip, who does 2-photon microscopy, he found that also the kind of crawling behavior of neutrophils is regulated by this CGRP, so that they're less active if you have the neural signal. So I think it's definitely regulating neutrophil function, recruitment, as well as function. And there's still a lot of questions there, how neurons regulate neutrophils.
So then we have Brian saying, incredible talk. Do we know what are the intermediate signals that mediate TRPV1 neuron response to streptolizing, or is there a direct interaction between SLS and TRPV1? That's
a very good question. So we don't think that the SLS directly activates TRPV1, partly because it doesn't need to, because it's already a pore-forming molecule. So it will punch holes in the membrane independent of ion channels. But on the other hand, TRP channels are known to respond and be amplified by increasing cations in the cytoplasm. So it could be that it may not directly interact with TRPV1, but it could then amplify the TRPV1 signal. So we had found in our study that heat-induced pain, I didn't show you the data, in that context was TRPV1 dependent. So it could be that streptolysin S helps set off a heat related signal that then sensitizes TRPV1 to heat-related signals. So the interaction between those two is still not clear, but I think that's something that's worth pursuing in the future.
Okay. Then we have JJ Sutachan, I think I'm saying this right, asking if microbiota, could microbiota regulate nociception transduction in cancer?
Oh, wow. That's a very specific. So I think we're very interested in whether microbiota regulates nociception. That's still, there's been a couple of papers saying that pain, or at least certain forms of pain, like chemotherapy-induced pain and things like that, are dependent on the microbiome. Whether that then regulates cancer is another very interesting question, whether nociceptors regulate cancers. So that's, the combination would be really interesting. So I don't know. I don't have the answer for either of those. But we are right now very focused on the first question: does the microbiome affect different forms of pain? And that would be interesting.
Cool. Yeah. Then, do you think the crosstalk between the skin cells, melanocytes, and neurons are somehow governed by the TRP channels in the context of pain and stress?
Right. So that study with Yachi, and it's a really, really cool study. I encourage you guys to read it. We found that actually the TRPV1 is not involved in the melanocyte stem cell loss. It's actually more of a trigger for the stress response. So what happens is the pain then leads to sympathetic neuron activation. So when you have a lot of pain, it induces stress. And then the sympathetic neurons talk to the melanocyte stem cells, and through ADRB2, which is the adrenergic receptor, and that causes them to change. So we've shown that because actually, if we just kill off TRPV1 neurons, that doesn't itself cause loss of the stem cells. So it's really the pain activation that induces stress that then induces stem cell loss. Yeah, but I think it does open the question of why you would couple that type of stem cell with neurons. It's a very interesting question, right? Like, what's the role of hair graying? You know, why does stress cause or pain cause?
Okay, so then what a thrilling story about gut. What would happen to the interaction between M cells and neurons in aging regarding physiology and or susceptibilities to salmonella infection?
So yeah, so you mean the relationship of aging with
what would happen to the interaction between the M cells and neurons in aging regarding physiology and or susceptibility to salmonella? So
that's a very good question. So aging is something our lab does not really study, but I think there's a lot of evidence that, you know, the nervous system does go wrong during aging. For example, pain goes higher, pain sensitivity goes higher in older people. And the innervation pattern of the skin, at least, I think, could change. I don't know much about the gut. That could be a very interesting idea if aging leads to dysregulation of either loss of neurons that innervate the gut or changes in density of nerves in the gut that could then feed into a defect, right, of homeostasis and host defense.
And then we have here a last one saying, fantastic talk and naive question. Is there any correlation between pain tolerance and infection susceptibility?
Yeah, so that's a very good question. So pain tolerance, like pain thresholds, is a hard to quantify thing, I think. But there is definitely a link between so like clinically, we actually have some data, but we weren't sure whether to put it into the story, but that like opioids, so like things that block pain, you know, there's very strong evidence that increases your susceptibility to infection. So I guess people who feel less pain or more pain, I would imagine that's reflective partly in these peripheral. So the thing is, perception of pain is somewhat different than not necessarily reflected by the neurons themselves. So you have to kind of separate those two. But if it's purely a physical thing, let's say there's more or less pain fibers in the gut, I can imagine that could lead to differences in host defense. But yeah, opioid treatment makes you much more susceptible to infections. So that's kind of fitting with our data.
Okay, so I think those are all the questions we have here. I think I went through them all. Now I just wanted to have a look and come back to our poll from the beginning. It's interesting to see that people engaged and that people voted on what they thought would be the field that they would position themselves. As you can see, we have here a lot of people from neuroscience, some from immunology, metabolism, neuroimmunology, development, and some that can't decide and like them all. But yeah, so regarding this, actually, I wanted to ask you, Isaac, where would you position yourself, and do you see yourself more as a neurobiologist, more as an immunologist, or if you find it difficult to find a place to fit your work? And
yeah, I just put myself down as neuroimmunologist. I have always been fascinated by the parallels and the interactions of these two systems, even from my college years. So I had been studying at that time the immune synapse, so to speak. So the formed by molecules in between immune cells, but the synapse coming from neurons. So I've always been fascinated by these two, and so it's hard for me to say I'm an immunologist or a neuroscientist. I think I love the brain. I love also the immune system. So
yeah, and I think, as someone was saying here in the chat, that there should not be those boundaries, right? And we should all look at these fields as interdisciplinary and look at the bigger picture, right? When we are trying to focus in something.
Yeah, okay. And I find it very helpful to see the different perspectives. And I love going to talks in both fields because there are somewhat different approaches. And I think bring people together in this type of venue is helpful in collaborations because people think somewhat scientifically even how you approach questions, from different fields. It's great.
Nice. Yeah. Very nice talk, Isaac. Again, thank you.
Thank you for hosting me, Raquel. You're a great host, and you did a great job. So thank you for all the questions. And
thank you. And I guess that's it for today. I just want to remind everyone here that we'll be back next week, same time, and with Professor Stephanie Schiermeier, who will be telling us about glia and glial neuron metabolic interactions in Drosophila. So I hope to see you all here next week as well. For the ones who registered for the Q&A session that we'll have just in a few minutes, you must have received an email with the link for the Zoom room, so see you there. And for the rest of the people, so thank you once again for coming, and I hope you enjoyed this as much as we did, and see you next Monday.
Wonderful.
So thanks.